So, yeah, I'm from the Texas Medical Center, Baylor College of Medicine, where we have about 7 million patient visits a year. So it's a large academic medical center like yours. And like you, we are focused on this question of how we can, this is a nostalgia slide, how we can bring science into medicine and achieve this goal of precision medicine that means different things to different people, but we, as I think as geneticists, believe that it means having a molecular insight into issues of diagnostics, prognostics, and care so that we can tailor treatments to individuals. That, of course, requires a deep understanding of the molecular architecture of disease so that we can put these pieces all together. And if you think about that ambition in the context of what we know about human genetics, now, after decades of both Mendelian genetics and GWAS studies, I think we can ask ourselves the question of how are we going to make the next steps in understanding genetic architecture generally, and hence the statement, can we build genetics piece by piece from the bottom up? Not as was been trying to be done really from GWAS, and do it from the top down. Now, this dichotomy, that between Mendelian disease, like cystic fibrosis and muscular dystrophy and all of the other 8,000 or so conditions for which are tractable, where you could find a mutation leading to a genetic disease with full penetrance, got the gene, got the disease, got the disease, got the gene, versus GWAS, where we have this abundance of signals across the genome, not really a whole lot of functional data underpinning that, and not a whole lot of actionable variants. It's well known to everybody here. So, like everyone in the field, we're looking for the Holy Grail, which is how can we bring about this complete molecular understanding of complex phenotypes, such as what kill us as adults, by understanding all the components. And if it's not here, we can't do it from this top-down view. And if this is too simple of a model, that the variants are invariably too penetrant, they occur too early, and they don't bridge into adult disease easily, where is the missing model? And here we label it oligogenics. Oligogenics is a catch-all word. It means you've got many different components of multiple genic contributors. But ideally, for each, and if you're doing a graduate project, for example, what you're going to do is you're going to find an example where there are multiple genes or multiple alleles that make individual contributions to a phenotype that in the right combination yield an adult disorder. We wrote about this recently, and I do invite the students at least to look at this manuscript because it tries to put in perspective the post-GWAS view of the complexity of Mendelian disease leading to adult disorders. So that's what I really want to talk about today, is that middle space and how I think we're actually making a little bit of progress. We haven't by any means knocked this problem out of the park. There's much to do, but the pathway forward is pretty clear. So I'm not going to talk very much at all about technologies. You all know this, that the genome's cost has gone down a lot, probably around $2,000 right now with the high-seqs, that exomes cost about $500 right now, and that still might go down further. We just got our first NovaSeq. This is the single color instrument. It's just running for us for the first time right now. I don't know if you've had a chance to work with it or seen the data from it, but if that comes into play and the real estate on it grows enough, we really could be down here in this very low dollar figure. Here's how the technology was applied by us in Houston, sort of along this pathway to where we are now. And as Stan mentioned, we're involved in the Human Genome Project and produced about 10% of that in Houston. And then since then, we did this series of individual personal genome projects that illustrate the principle that's put us where we are today. 454 was great because we got to sequence Jim Watson. They generated the data there, and we gave him this data in Houston. You can look at, if you're nostalgic for that, I can't imagine why you would be, you can look on YouTube and see Watson taking his large disk, hard disk with his 454 data on it as his personal genome. We did it that way so that he would have all the liability. We gave him his data. He put it, the Cold Spring Harbor people put it on the web. This was a great feel-good international project. Desmond Tutu has more Bushman in his background than he thought. James Lubsky, a few years later, we sequenced him and found his Charcot-Marie tooth allele that causes a disease in him. You know, if those of you know who Jim know that he's an expert on Charcot-Marie tooth, and he's found many of the genes that cause that disorder, but hadn't found his own after a decade or so. But we found that in what to us was spectacular then, but now we all take for granted in a couple of weeks of sequencing. These children here, you'll see on the Baylor website, because they remain the principal example of children that got a pill as a result of their sequencing that completely cured their condition. So unfortunately, a very few of those. And I could go into that story in more depth if you want. And then this was sort of a whole genome period with primitive whole genome technologies. We developed the exomes and got into discovery through de novo mutation events, found our first research cases of these disorders, and then used that as the basis to launch a diagnostic laboratory at Baylor that was a collaboration between the genetics department and the Genome Center. And in 2011 opened up a clinical service that I'll just mention again in another couple of slides. And this here has all been the underpinnings of our work that's formed our thinking about human genetics. Over here I've got what you might regard as cheesy, but in Houston it's taken very seriously, I can tell you, an analogy of the space program with V2 rockets up here, a little bit of an improvement going to the moon, and now and there's a space shuttle there. Later I'll challenge you with what the ingredients of the Mars program should be if this was the Moonshot. So what I'm going to do is just talk a little bit about the whole genome extensions from this in a couple of slides and tell you what we've done in the adult space on the right-hand side of the genetics graph, the things with large cohort populations, trying to do top-down solutions for adult genetic conditions, and then I'm going to jump back into the Mendelian story and just talk about that then for the rest of the comments. So here's the whole genome adult story. I work with Eric Bowinkle. He's at the University of Texas, and he's accrued a number of cohorts, and we work through him on a number of the national ones. This is the atherosclerotic risk in the community. And the basic idea here is to bring individuals into the collection and give them clinical level phenotyping in multiple visits, and then monitor their various parameters, and then to perform a genomic analysis, and then to perform association studies looking to mine out those conditions. And here's two examples of where that's actually worked out pretty well. So here's one where we're looking, postdocs looking at hematopoietic pathways, has looked at different parameters related to the steady state numbers and representation of these different cell types, and then gone ahead and identified individual marker that in one case corresponds to steady state platelet levels. If you're a carrier for this variant and it replicates in another population, you actually have lower levels of platelets. And that phenomenon is reproduced if you go into a cellular functional study and engineer that marker into the cells, and it fits in with the whole pathway and can be rationalized as having ultimate physiological impact. The gene for which this marker exists, this is intronic, but the gene where that exists has other cases where there are severe Mendelian cases of mutations that dramatically affect platelet function. So here we have, by mining a large cohort, a process of association which brings us down to a single gene, and a marker with not too low a frequency which influence platelet levels and potentially relates to human disease type. Here's a methods that we developed about that time, which was to window along genome data and to, in a more sophisticated way, look to improve the annotation procedures by looking at regulatory mutations in regulatory domains and in other parts of genes, but basically with better informatics tools. And then correlating that data with amino acid data that became possible because of more sophisticated analytical phenotyping procedures that came into play as the cohorts grew. And the outset of this story is that, there's actually a number of these kind of stories, but this one I'll tell you about is that one gene, which is involved in asparagine genesis, has in some cases profound mutations that affect its function, but here, interestingly, a common variant which influences the level of that gene, which downstream is correlated in frequency associated with, in the different genetic states, different levels of production of arginine. So what am I telling you? Well, just two studies here where we look at cellular levels in the circulation, or in this case amino acid levels, and there's multiple ones of those. We're mapping out these endophenotypes really with quite good precision. That's great if we're interested in endophenotypes, but the link from there to human disease is actually a little bit thin. Right? We don't, in these cases, have strong associations with disease endpoints, and that's the criticism of the whole approach. We would say that these somewhat impact disease knowledge, and that knowledge is growing. So this is good, but this is our statement about the state of cohort mining in large populations with large studies. So let's turn away then from the genome studies and go back to exome studies that we've pursued more in these high penetrance, clearer phenotype cases. Exomes, of course, I have to say, this slide I first made in 2007 or 2008, because as soon as we developed the exome methods, people came up and said, they're not going to last very long. That whole genomes will take over, right? And that question gets asked pretty much once a week. The exomes are still remarkably powerful, of course, in the bang for buck way, and they do maximize discovery of all kinds, even though if it's a case you're particularly interested, that might not work for you. Here's the other technical slide I'll show you as we go into the exome story. We've actually worked a lot to refine the exome reagent. We worked with Nimblegen all through our history of this and developed a reagent that's called V-Chrome. And now, because of the missing coverage in genes that are part of gene tests that are in the diagnostic lab, we've developed a series of supplementary oligonucleotides that fill out some of the original genes and add a couple more in and increase the space. So how good is this thing? Well, if you compare it to the best standard, which right now is this color here, this is the PCR-free best enzyme whole genome data that we've got. And you ask, of the genes that you're interested in, that you've ever attended to and worried about their coverage, how many of them are covered completely? That's the number from the best whole genome. This is the number from the supplemented exome. So in other words, for the regions you're interested in, the exome are pretty damn good. So those manuscripts you read that say that exomes aren't so good as genomes, look carefully at the quality of the exomes before you draw your own conclusions. So what have we done with exomes in this Mendelian space? This summarizes it all, and so I'll just go over this slide and take a moment. As I mentioned, we started that clinical lab in 2011, and it's actually been sold by Baylor College of Medicine, which is the institution we work in, to a Japanese company. It's a partnership, and the new lab is a commercial venture called Baylor Genetics. So that group has maintained about 200 to 400 cases per month, and this, I'm not sure what the time is here, but it goes on and on since it grew in 2012. There are about probably 14,000 cases gone through there now. Now, here's the situation. Unlike a research lab, where you have to go and bang on the door of somebody and say, do you have families we could study in order to study Mendelian conditions, the beauty of the clinical lab is that cases come prepared. They get sent to your door. And in every one of these cases, although not in every case that comes to the door, insurance companies pay for the first pass of the analysis. So you get genetic data, you get a patient, and then this is beautiful material for the research arena. Now, of course, there's a big hurdle here. You can't just take these patients and put them into the research arena without adequate consent. So much of the work we've done is to try and streamline this process where the unsolved cases from the diagnostic clinic get pushed over into the Mendelian studies, into other discovery engines. The solution rate here is about 25%, and that's a controversial figure, and I'll come back in one slide in a minute just to address that. But for now, the 75% of the undiagnosed are the kind of the research fodder here for our Mendelian program and other individual investigators at the institution who like to take these families and solve them. So they're doing pretty well. This is the progress report slide I just took from the Mendelian guys yesterday. And this is designed to show you a lot of genes that are large in number. There's 219 discoveries from the Baylor Hopkins part of that program. And then hundreds more, which are either should have been diagnosed in the first place, and in here, this is called phenotype expansion. So phenotype expansion is when you find a gene and you say, aha, this is a new syndrome, but somebody else says, you should have been smarter and figured that out in the clinic, right? And then when they report their things, you say, well, you should have been smarter too. So a lot of these are actually probably justifiable as novel discoveries if you lowered the barrier for what's novel and what's not. So hundreds and hundreds. It's an interesting curve. What's what underpins this is how is the process of Mendelian discovery going? How, where's it going to end? Are we going to find 8,000 genes? And I think what you can see here is on the novel genes, it's kind of going up and up and up. So who knows where it's going to end, but it's certainly not flattening out yet. We're still on the discovery ramp. Much of this discovery has been enhanced by sharing of preliminary data from all sources with other investigators and across the local network. And so here's a published example where some postdocs have taken 80 cases or so, and for each one they have reached into the Centers for Mendelian Genomics. This local clinical lab, WGL, is our first iteration of the clinical lab. WGL is the second iteration, and Gene Matcher, which is this online computer service you can go to and put your gene in and find other people. And in each case, it's been an assemblage of multiple individual cases and families through that's led to the elucidation of a gene underlying the particular phenotype. So that's good stuff. Gene Matcher is going great. This is our local input, but if you go to Gene Matcher and to others will deliver you the statistics, then you add to this nationally and internationally, the numbers of things going there is terrific. So I urge any of you working on a gene to be not shy about putting your gene name into that resource, because you will find people that you can then choose how much you're going to work with to follow up. Okay, so here's the output of that clinical lab, and on the clinical side, not the research side, that I said had a diagnostic rate of about 25%. This is very often challenged. Others come and say, hey, we've got a 30% diagnostic rate, or we've got even a 70% diagnostic rate. And so we turned around and looked at all of our data. Some of this is on the research side, of course, but above line is the clinical line. And asked ourselves what different ways can we produce a rate measurement. And it turns out, as you'd expect, it's usually the denominator, not the numerator, that defines the difference in that equation. So this stuff here is really characterized by people who come to the clinic just because there's a suspicion of a possible problem. That's the broadest net you've got so far. And as you move down the graph here, you'll see the numbers get higher because the samples have been filtered or curated, or somebody's bought their collection, or this kind of thing. So you've really got a richer set. Maybe there's more familial evidence. Maybe there's partial molecular data. Maybe there's more homogeneity in the phenotype. All those things bringing a higher rate. So we keep these numbers in our pocket, so when somebody says they've got 70%, we say we get 90%, and it's always a great conversation. But what I like to do, really, for the main substance of my comments, is work through three things that we've derived from the sum of all of these Mendelian studies, in contrast to those earlier complex disease studies where we're able to just mine out these more weaker endophenotypes. I want to talk about how the allelic complexity can be realized by burrowing into some of these cases. I want to talk about how multilocus and multi-allelic conditions can be revealed simply by the Mendelian screening, and show us how you can get real complexity out of the Mendelian stories. And then take that then and leap over to this oligogenic space and burden models where we have early data that says that we can really truly bridge Mendelian and adult studies. And I'll refer back to some of these studies as we go. So here's this first question: can we see enough allelic complexity in some of these cases? And here's two examples. This is one disorder that was early on our list. It's a gene for which very little is known, the AT hook DNA binding motif containing protein number 1. And we started with 4 patients that had de novo mutations. They weren't linked phenotypically. In other words, you couldn't take them all and say they belonged in the same phenotype bin. But once we had the de novo mutations, it was pretty obvious they all belonged together. And so we developed an understanding of that disorder. Now, it's autosomal dominant, and the mutations are truncating, and they all occur in this critical region. But it leaves alive the question of what kind of other mutations could occur in a larger population, and what is the exact molecular mechanism. So we're burrowing away into that now, studying the question of, is it haploinsufficient or dominant negative, which will tell us something about the fundamentals of the molecular mechanism. But what's particularly caught our interest is the report. We're up to about 30 families now, because we have a registry, we've been reaching out, Facebook page, all of that good stuff. We have a missense de novo mutation in the critical region that was called in a boy who is teenager autism. All of these other mutations, all of the early patients, profoundly affected, very young, no speech, cognitively very low level of operation, It's quite a devastating disorder. But this missense case turns up with potentially autistic. Here's a case where there's a frame shift that's upstream of the critical region, and this is wrong. This is actually in-frame deletion, multiple amino acid deletion. It's upstream of the critical region, though, and it's a 63-year-old man who is described as mentally retarded. So here's the underlying question. It's not a new question in genetics for sure. That is, can mild mutations give mild phenotypes? But here's an arena to apply it in that's actually quite new and quite fresh, because we're discovering anew all of these de novo mutations in neurological disorders. So it's kind of a new chance to burrow into this issue of phenotypic complexity and allelic complexity at single loci. Here's the second example. It's almost a mirror of the first. That is, that there were 4 cases that were found that linked truncating mutations, de novo truncating mutations in this case a transcription factor that gives rise to a very severe phenotype. And if you look across the gene map here, please don't go crazy trying to look at the different, all the different letters, but you see the colours. The green lines here represent mild cases, and the red represent severe cases. So we were left, and there are also some cases of, there are some instances of truncating mutations in the gene in public databases where they shouldn't be if this was a profound case. So we were left there with the question of how can we really explain this if there are simply unexplainable so far truncating mutations. Well, fortunately, we've been working with some guys at Regeneron who are listed here. Claudia was a graduate student at Baylor, and she moved there, and we worked with the rest of the group. In a nutshell, what they've been able to do is to rebuild all these genes in different models and look at nonsense mediated decay and nonsense mediated decay escape, and they've shown pretty conclusively that the mild mutations all have message decay so that the damaged allele goes away, whereas the profoundly affected individuals, the message doesn't go away, so it likely produces a truncated protein and gives a dominant negative effect on the phenotype. They've done this in a cellular system, and they're moving that up now to a mouse system. So 2 examples there where the single locus burrowing into it can now give good insight into molecular mechanisms, their heterogeneity, and potentially phenotypic heterogeneity that can be downstream from that. So this is great, good genetics, good science. It exemplifies what happens at these single gene studies, and that looking at the phenotype carefully and at other omics studies can really enhance our knowledge, and it reminds us all about phenotypic complexity. But it's just one baby step, I would say, towards that oligogenic space. So here we are in that second of the 3 things to tell you about. This is the idea of multilocus models. So as we started to ramp up our cases of routine clinical diagnostics of the Mendelian cases of children coming into the clinic, someone saying, we think there might be a Mendelian problem here, we just can't figure it out. A surprising finding was that 5% of the diagnosed cases actually had 2 Mendelian diseases within the same individual. Now, I've got to say that you guys here found some of these as well. Nobody else in the whole, like across all of the early Mendelian studies, seemed to report these dual cases. So I think it's a tribute to you locals that you also went in and burrowed into these cases thoroughly. Some of the individuals had 3 cases of Mendelian disorders, and one even has 4. So these are pretty interesting. So Jennifer Posey here, one of the clinical fellows, has burrowed into this story to look at how this complexity arises, how we can map it and use it to get into this oligogenic space. And she's taken this approach. First of all, it's not just looking like the familiar modes of digenic inheritance or multigene effects. So we know all about second hits in cancer. We know about models where there is 2 genes are necessary to drive the disease, or even 3 when there's multiple alleles and multiple genes. This is something a little bit different, and it can be passed out into 2 sort of conceptual areas. One where there is a distinct phenotype, that is that there's 2 genes and each gene gives a separate phenotype, but when the individual presents with both phenotypes, you can't recognize the individual. Or second, where the phenotypes kind of overlap, and so you get some really compounded phenotype that is also unrecognizable. So she's been asking about this question. Here's examples. Here's some of you may know these syndromes well, but here's basically a case where one of the genes, most of the phenotype is associated with microdontia with teeth stuff, and the other is a skin problem, right? So you're not going to mix up those phenotypes if you see them separately. But in the second case, both of the disorders have phenotype characteristics that are expressed in the skin and therefore compounding the whole model. So this is the separate and this is the blended. And what, well, the first thing Jennifer did is ask were there any obvious compounders for driving this kind of thing? Maternal, particularly paternal age, parental age, particularly paternal age, is a driver of many of these things. No apparent effect there. There's no particular class of mutations by either nucleotide content or autosomal dominant or recessive, etc. There may be some under occurrence or ascertainment, and when the statistical analysis was done, comparing those cases where there's just one gene causing the phenotype versus more, it seems to be there's an underrepresentation of those that are there more often. Now, the way of generating these statistics is something we could talk about at some length, but that's the summation at this time. It's actually a little counterintuitive, but we can come back to that. Anyway, what Jennifer and colleagues did was look through all of the cases and make the clinical geneticist assessment of whether this is a blended or a separate phenotype, and then establish a score, declare that as a binary function, and then separately take the phenotypic terms that could be taken out of the clinical records, and they're making this story very short, crunch that through a series of software that uses ontology trees and the clinical data and then computes a score based upon where the clinical data maps in the ontology tree. And this score then is meant to represent how closely aligned or different the phenotypes are to known Mendelian disorders. And the good news is that the humans and the computers kind of agree. The humans, of course, give a binary output, distinct versus overlapping, and the computers, the distinct and overlapping versions of the phenotypes are separable. Okay. So this is good stuff. This means there's computer programs here that can look into these complicated Mendelian phenotypes and build stories of complexity, which is a bridge towards our state of oligogenics. Now, some of these genes you can burrow into, some of these gene combinations you can burrow into and correlate the degree of overlap and the degree of separateness with a molecular model. Do they appear in the same hairball of functional interactions? And that too can add to this whole story. So I'm summarizing all of this now, that these dual molecular diagnoses that occur in a significant number of the exome diagnoses are real things. That means diagnostically you shouldn't ignore them, and they can lead to complex phenotypes. So the real direction this is pointing into is that a subclass of these blended phenotypes may reflect some kind of a genetic load that their interaction looks like a complicated enough phenotype to call it a common disorder. And that this is a sort of baby step to that space of oligogenics. So if you want to be more direct, though, and study oligogenics, then you might want to take a slightly different approach in looking back at all of that data. What you should do is look at those cases where you have Mendelian solutions and ask yourselves, what's going on in the cases that you can't solve with a simple Mendelian methodology or a simple Mendelian explanation of the genomic data that you derive. So you go find that syndrome, and then you identify all of the known disease alleles in there, and this, of course, will find you all of the Mendelian cases. We'll put them aside, and then you look at the rest and say, what else is there, and you'll try and fit genetic models. Now, ideally, in a proper design study, you do this in bulk with a lot of samples, you would come back and apply a quantitative model that would do multi-allele tests across the whole genome and look for any variable that associates between any pairs of genetic variables that associate with the ultimate phenotype. Now, given the vast difference between the number of samples we've been able to work with in these Mendelian studies versus the whole genomes and the research cohorts where we're dealing with thousands and thousands of thousands, we also have to be prepared to work with more qualitative models where we either leverage this understanding with functional studies, or we use some other kind of clever statistics. So that's the background for this third part for looking at oligogenics. Now, until very recently, this was our poster child and our only story in that space. we went and looked at Charcot-Marie Tooth that I mentioned earlier was the disease, or is the disease, in Jim Lubsky, for which he studies, has studied for many years, where in the old days it was thought to be one disorder, but the molecular studies soon began to dissect into many different loci, and there are now about 50 genes for which mutations can be the explanation of a Mendelian story explaining Charcot-Marie Tooth in the familial cases. Now, like many disorders, many of these syndromes, Charcot-Marie Tooth has these familial forms for which we have that explanation, but they only represent a couple of % at most of all of the forms. Most are sporadic. So we're really looking now in this, what's the Mendelian, what's the common disorder story, and now we're looking at these unsolved Mendelian cases, these ones with some familial hint to begin to build that bridge. So when we did that, and the numbers, like I said, are not great, not large here. There was 37 families where we had them as unsolved. And there's Claudia and Tamar, Claudia before she went to Regeneron, sequenced these families. In a couple of them, they said, oh, we should have found the gene, so that brought the numbers down. But then in most of the others, they found a surprising number of variants in genes that looked like they were the Charcot-Marie Tooth genes, but none of them on their own, or few of them, could make the full Mendelian story. Okay, so this is the idea then that if you have multiple genes involved in the same process, if you have a carrier mutation in one and a carrier mutation in other, in the same person, in aggregate, that might give you a contribution to the phenotype. It's a genetic load argument. It's a misuse of the term genetic load, but it's what we call genetic load right now. So here's the data, taking all of those families and doing, looking at controls. You see the number of rare variant alleles that accumulate in the cases is larger than in the controls. We have a difference here. Now, we couldn't get that genome-wide with 37 genes. To do that experiment statistically, it was necessary to restrict the space to a subset of genes that could be, frankly, gave the effect. But the experiment was to take the 400 or so neuropathy genes and look in that space. So we didn't just look at the genes that had the variants. We looked in a set of genes that conform to a functional set that should be of interest. Then we looked at the mutation data. Then we said we've got an excess burden. So this is pretty exciting, and that's statistical, a statistical analysis. And we fed those individual variants over to Nico Katsanis, and he built different combinations of these alleles, the ones that were observed in the individual cases, into the fish model and measured the fish phenotype, and it's all catalogued here. And the summary of all of these, all this histogram here is that you can recapitulate the burden model that you observed statistically in the Charcot-Marie Tooth cases in the fish model. In other words, if you have an accumulation of alleles in a person, you put that in a fish, the fish looks more severe than if you don't have that accumulation of alleles. Now, got to be clear about this. In the original burden study, in the 37 families, this wasn't a case of the person had no evidence of any Charcot-Marie Tooth causing locus, except for this smattering of carrier mutations. The predominance was of cases where a single locus explained, with some confidence, the Mendelian story in that individual, but the phenotype was greatly exacerbated by the presence of the additional mutations. Okay, so we thought this was step one. This is not the poster child. This is just the beginning of this whole long story we're about to embark on. So we did a whole series of these kind of things, running out, quickly broadening our net of what we brought into the Mendelian space to study, believing that we would soon find a number of other examples with this same kind of carrier space story. So we started with a cerebral palsy cohort that was collaborators in Adelaide, Australia had, and they had nearly 200 families. So cerebral palsy, of course, is not supposed to be genetic. It's supposed to be just anoxia at birth. But in these families, there was actually, in many cases, some evidence of a familial pattern. So really clearly something going on, and also a more profound molecular case. So we went and sequenced the 100 or so there, and we did indeed find a number for which there were clear genetic causes and some new loci that were involved in those developmental disorders, but nothing that looked like oligogenics, nothing that survived that same kind of analysis. And then the same story in this semi-homogeneous collection of Turkish families who were brought to us by Ender Karaka, a fellow. Similar numbers, similar process, sequence everybody, and look for the genetic cause. We found those in many of the families, many known genes. We found a number of new candidate genes, nothing that looked like oligogenics. Arthrogryposis, same kind of story. Actually, a little more promising. In the 48 families, there were 17 with at least one mutation, and a few of those that seemed to have two loci that were involved. So this is what we call this, a smattering of oligogenics, and looking like it. And then the final one, we're working with Josh Schulman at Baylor. He's at the NRI, the Huda Zogby's new Neurological Research Institute. He had looked at, with us at 150 cases of Parkinson's disease, and in fact found two new candidates that have since borne out, and shown the genetic heterogeneity. At that time, nothing at all that looked like oligogenics. And if I'd talked to you 2 weeks ago, that would be the end of the story. But I was telling Josh this story and at the coffee shop, and he said, oh no, we took that into the National Parkinson's Consortium, and we're now up to 1,200 samples. And now we see a burden model that we can explain that this looks a bit like that Charcot-Marie tooth case, where the number of sort of interesting looking variants per individual is greater in the cases. This time, now this is still not genome-wide, but this time he's taking it across the lysosomal storage disorder gene set. Okay? So there's a reason to associate that class of genes with Parkinson's, but if you take the broader set of those genes and then run this analysis, you see the burden model. So the story here is pretty simple, that if you take these unsolved cases in these syndromes and you generate the genomic data, and then you increase your numbers to something approaching a level of respectability, then you can begin to see these signals. It really does invite us to boost the numbers and do proper power calculations. Now we've got a bit of a handle on the genomic geography of this, and look carefully at what these loci are and how they make the different contributions to the overall phenotype. This to me is extremely encouraging. And I heard a nice talk from Steve Chanik about heritable cancers, talking about an osteosarcoma study. Same thing. It's getting large enough now, heritable factors leading to osteosarcoma, satisfying a burden model in a restricted gene space. Okay. So this is like the punchline of my talk. How do we do that? So we're showing that the deep analysis of these unsolved cases can show a genetic burden. If you can do functional studies like we did in the Charcot-Marie tooth, you can validate that model. All of these are way underpowered. We need larger samples to get into this space. Now, that begs the question, how are we going to do that? And I think one thing that we would, as a community, us at Baylor and you here, would agree on very quickly is it's hellishly difficult to get into these larger sample numbers by continuing the model of banging on doors and looking for research samples. The only way to do it is to go into clinical catchment areas. And that's, in fact, when we, in Houston, look back at our clinical diagnostic service, what we've learned from that, the thing that we have mainly learned is that the way to satisfy the feeding of these larger genetic studies has to be through the clinical portals. And the imperative then is to bring down the barriers between the clinical portals and the research cohorts to get the full fluidity between those spaces. And so the work that everybody's doing to enable that, of course with full patient protection and all that good stuff, is of critical importance. We need to boost those numbers. Now, how do we do it? Now, and who do we do it in? Well, we should be doing this in adults, of course, not just in these pediatric cases that are unsolved. And this is just what I said, we need the clinical integration. Now, to help achieve this locally, we build a large data warehouse, data lake, data island. These are buzzwords that we're all working with, and I know you have a similar effort going here. This is the idea that if you want to achieve this magical integration of the clinical space and the research arena, you've got to somehow bring together all kinds of data in a way that can work with the data and make queries work across different formats, maintaining levels of privacy, etc., and now allowing you to use tools on this kind of data without everything being too cumbersome. The data sets are huge. It's all the problems that we talk about in big data. So I'm betting you've got a diagram like this here, where your medical record and your limbs and different levels of patient and research data all push into the data lake with variant data, sample data, phenotype data, and that individuals are able to analyse those and researchers can pull from them for their analysis. So I'll tell you what I do most of the day when I'm not doing email and grants is I sit around talking to people about compliance levels and computer architecture. Does this sound familiar? That really the question is, who can see these data and at what stage do the data become revealing of the different levels of compliance, in violation of different levels of compliance you need for research protection and for HIPAA. And so how do you apply that encryption? It's a big problem, but it's one that we're all progressively solving in step. And what's great about this is that the whole thing is worthwhile doing. So here's this is actually a few weeks old, this query. Actually, I'm lying, it's just before the turn of the year. So we finally got our database together and we pushed into it every VCF that we could get our hands on from the Genome Centre. It included all of the Mendelian cases in the research and the clinical arena, and it included reference cases from our local cohorts, and we pulled in EXAC and 1,000 genomes, etc. At this time, we had no clinical data at all, no phenotype data. But we asked ourselves the question, could we begin to mine this data for interesting variants and for gene disease clues that we could then take into the phenotype data for validation? So we put up a simple query. Let's take everything that's not already known and look for loss of function and set filters for the frequency, whether they'd ever been seen before and what EXAC says about them, and search across there. And out dropped a bunch of candidates, and I called around the centre, and first of all, three of them were actually just published, but not in OMIM, right? That's a good sign. It was like a blind test. And then several of these other genes were already being worked on by different postdoctoral fellows and clinical fellows who already had sort of private access to the data and had an interest in that general area and already picked on that gene. This is like tremendously encouraging, right? Now we've repeated this not with the loss of functions, we've also repeated this with missense. We have a similar large candidate list and we're pushing that out into the discovery space. So this is sort of good news, the data works. So here's the future for us. And people ask me, what are we doing in the Genome Centre and why do we exist? I tell them, well, doing the pediatric studies and getting it so that we had routine collection of the Mendelian data from these pediatric cases and then pushing that data over into the research arena been really useful. We call that the Moonshot, and we no longer want to build houses on the moon. We want to go to Mars. Mars is solving this in adults, and it is doing the same thing in adult clinics. So we're bursting to get our methods pushed into adult clinics where we routinely collect the data and make it available for discovery mining without necessarily being heavily hypothesis-driven and front-loaded with what we think we might find. That, to us, is our Mars mission. So that's the end of my comments. I really thank you all for listening. I guess I've gone quickly. I finished 5 minutes early here. I want to thank all of these guys. You may know Donna Musney. She's the operations officer in the Genome Centre. And Eric and Jennifer I mentioned. And I think some of you recognise all of our colleagues here. But thanks a lot for inviting me. It's been a pleasure to come and to meet people today.